Since the first report on Alzheimer's disease in 1906, we have made great advancements in our understanding of the disease. We've got detailed molecular mechanisms explaining the characteristic pathology, increased aggregation of beta amyloid and tau protein in the brain, leading to neuroinflammation, cell death, and brain atrophy. But despite these advancements, we're still pretty much clueless about the driving force behind this pathology. In this video, I argue why I think that impaired brain energy metabolism is the driving force behind Alzheimer's disease, and that the pathology that we see is actually a mechanism evolved to protect our brain cells against infection. So this is not about the small percentage of cases in which genetic mutations lead to an early onset of Alzheimer's disease, since we know the driving force in these cases. Mutations in the enzyme that cleaves beta amyloid peptides from its precursor or in the precursor protein itself leads to a relative increase in the production of longer beta amyloid peptides. Longer peptides aggregate more easily than the shorter peptides. Think of hairs in a shower sink. The short ones easily get cleared. The long ones aggregate and plug the sink until somebody is brave enough to take them out. Recent research has shown that the mutations leading to the biggest increase in long amyloid peptides are associated with the earliest onset and the most rapid progression of Alzheimer's disease. But again, in absence of these mutations, we do not know the driving force behind this pathology yet. Epidemiologic studies have provided risk factors for Alzheimer's disease. The interesting thing about common risk factors is that they give away clues on the driving force behind the disease. What do they have in common? So logically thinking, it seems that all the risk factors impair the cerebral energy metabolism. If the blood doesn't circulate properly, that limits the supply of nutrients to the brain. Low mental and physical activity, depression, and traumatic injury limits the brain's use of energy, and diabetes limits the transport of nutrients from the blood into the brain cells. And of course, aging is involved in all of these processes. And actually, there's evidence for this. Here, aging people get a PET CT scan. We report a significant correlation between aging and the decrease in global brain metabolism. What about diabetes? This review highlights the role of impaired glucose metabolism in the pathophysiological cascades of diabetes. Glucose transport, glycolysis, the pentose phosphate pathway, and the TCA cycle in glucose metabolism are aberrant in the brains of diabetic patients and animals, leading to reduced ATP synthesis, the energy unit of the cell. A study on late-life depression. The depressed subjects showed widespread reductions in the regional cerebral metabolic rate for glucose in most major neocortical, subcortical, and paralimbic regions that were significantly different from control values. Traumatic brain injury. Traumatic brain injury evokes a prolonged glucose metabolic depression and consequent energy crisis. What about this recent study on cardiovascular disease in The Lancet? Persistent high risk of cardiovascular disease was associated with an accelerated decline of cortical FDG uptake, a glucose marker, compared with low risk. Moreover, progression of subclinical carotid atherosclerosis was associated with an additional decline in FDG uptake in Alzheimer's disease brain regions. So this one is a total no-brainer, but anyway. Neuronal activation requires increased metabolism, so ATP generation. That means that low mental activity means low metabolism. What about physical activity? There were associations between moderate physical activity and glucose metabolism in all regions of interest examined. In this case, the regions known to be hypometabolic in Alzheimer's disease, so the hippocampus, posterior cingulate, etc. So we know that people with Alzheimer's disease risk factors tend to have impaired cerebral energy metabolism. The next important question is, does impairment of cerebral energy metabolism actually lead to Alzheimer's disease? So obviously there are no studies investigating this in humans due to ethical reasons. Or maybe there are, who knows. But there are several studies testing the impairment of brain energy metabolism in animals and in cultured neurons. These researchers impaired glucose transport in mice brains by reducing the expression of a glucose transporter. Glucose transporter 1 deficiency in mice overexpressing amyloid precursor protein leads to accelerated amyloid pathology, reduced amyloid clearance, diminished neuronal activity, behavioral deficits and progressive neuronal loss, and neurodegeneration. In this study, researchers limited the supply of energy to the brain of mice by inducing carotid artery stenosis, mimicking cardiovascular disease and atherosclerosis. Chronic cerebral hypoperfusion increased amyloid fibrils and induced amyloid deposition. Next one. Energy inhibition by blocking the metabolism of glucose in mice. Reduced energy production caused an approximately 150% increase of cerebral base 1 levels compared to controls. This is the enzyme regulating the production of beta amyloid. Cerebral AB40 levels were increased to approximately 200% of control. So what about tau protein? Compared with controls, mice with brain glucose deficits showed significant memory impairments, reduction of synaptic long-term potentiation, and increased tau phosphorylation. This is the state in which tau aggregates instead of supporting the cytoskeleton of the cell. Another one on tau, but this time in cultured neurons deprived of glucose. Compared with controls, cells incubated in the absence of glucose had a significant increase in tau phosphorylation. Now, what about beta amyloid in cultured neurons? Energy deprivation induces processes which increase the translation of base 1 and elevate amyloid production in primary neurons. So it seems that decreasing the energy metabolism leads to characteristics of Alzheimer's disease. But what about interventions that increase energy metabolism? In this study, they enhanced glucose metabolism by upregulating glucose transporters in the brains of flies expressing beta amyloid. Our study has shown that experimentally increasing glucose uptake in neurons can protect against amyloid toxicity. This is a recent meta-analysis on the effect of intranasal insulin on cognition. Insulin can enter the brain to the olfactory nerve and increase the transport of glucose into cells that express glucose transporter 4. Patients with Alzheimer's disease or mild cognitive impairment treated with intranasal insulin were more likely to show an improvement in global cognition. We just saw that mental and physical exercise increases brain energy metabolism. Studies have shown that elderly who regularly engage in mental and physical activity have lower risk of dementia development. Complex mental activity during life is associated with a reduction in hippocampal atrophy rate, while regular physical exercise can slow down the progressive reduction of the cortical brain volume which occurs during aging. So again, we know that people with Alzheimer's disease risk factors tend to have impaired brain energy metabolism. We know that in lab studies, inhibiting energy metabolism leads to Alzheimer's disease pathology, and increasing energy metabolism protects against it. In humans, mental and physical activity protect against Alzheimer's disease, and they increase the brain energy metabolism. So, its role in Alzheimer's disease is undeniable. An important question to ask at this point is, why? Why would energy impairment in the brain lead to Alzheimer's disease? And I believe that there are pieces of evidence that give us a clue why. And for that, let's take a look at one of the most ignored lines of research in Alzheimer's disease, the infectious hypothesis. This hypothesis claims that infection is the root cause of Alzheimer's disease. So first of all, I do not agree with this claim, simply because not all people with Alzheimer's disease have brain infections. But there are interesting findings in this line of research that may give reason to Alzheimer's disease pathology. First of all, amyloid plaques have been shown to contain microbial DNA. In this study, researchers show that in Alzheimer's disease brains, 90% of amyloid plaques contained viral DNA, and 72% of the DNA co-localized with the plaques. These researchers infected cultured brain cells and mice brains with a herpes virus. Infection of cultured neuronal and glial cells with HSV1 leads to a dramatic increase in the intracellular levels of AB40 and 42. Similarly, AB42 deposits are present in mouse brains after HSV1 infection. So beta amyloid is produced in response to infection, but does it actually exert antimicrobial activity? These researchers tested AB peptides of different lengths on different pathogens. AB42 exerted an antimicrobial activity on all tested pathogens, killing up to 80% of all microorganisms within 6 hours. AB40 only had a moderate antimicrobial activity against Candida albicans. So the longer peptides seem to be more potent at killing microbes compared to the shorter peptides. Now, what about tau protein? So there's actually no published research on this to my knowledge, but I found this on the research page of the Cure Alzheimer's Fund. Our preliminary data already suggests tau is a potent antibacterial. So even though not all patients with Alzheimer's disease have a brain infection, the characterizing pathology seems to have strong antimicrobial effects. To understand this, I believe we should look at the evidence from an evolutionary point of view. Is there an evolutionary advantage that can explain the protein aggregation and subsequent immune response seen in Alzheimer's disease? Take a look at this diagram at the bottom. Alzheimer's disease is a post-reproductive age disease. This means that it does not influence evolution by natural selection, since this happens before and around reproductive age. And vice versa, evolution does not take into account the process of Alzheimer's disease, since it happens way after reproductive age. We know that the amyloid precursor protein is around 600 million years old. Metabolization of these proteins has literally had millions of years to evolve and fine-tune itself. So maybe the process of protein aggregation and neuroinflammation that we see in Alzheimer's disease at old age is actually beneficial for survival up to and around reproductive age. Now, we know that microorganisms use the energy sources of the host cells that they infect. What is this? Pathogens, including viruses, bacteria, fungi, and parasites, remodel the metabolism of their host to acquire the nutrients they need to proliferate. Thus, host cells are often perceived as mere exploitable nutrient pools during infection. Radical, right? So, for illustration purposes, here is a brain cell that relies on glucose for its energy metabolism. If this brain cell gets infected by a pathogen and the pathogen starts using the glucose for its own good, what happens to the energy metabolism and the energy availability of the cell? It decreases, of course. So let's combine these pieces of evidence to see if we can find the missing link. We know from lab studies that impairing the energy metabolism in brain cells leads to Alzheimer's pathology. Infection of brain cells also leads to Alzheimer pathology. We know that pathogens feed off the energy supply of their host cell, and also note that they're very good at hiding themselves from the immune system. And keep in mind, the brain's immune system is limited compared to that of the rest of the body. The blood-brain barrier prevents all kinds of harmful substances from entering the brain, so pathogens, but also specialized immune cells. Due to natural selection, the brain cells may have evolved to interpret this decrease in energy metabolism as infection, because at reproductive age there simply aren't many other factors limiting the energy metabolism this way. So let's put this idea into practice in a reproductive age brain, so how evolution probably intended this mechanism, versus in an aging brain with Alzheimer's disease risk factors, so what evolution did not foresee. In a reproductive age brain, when a pathogen infects a brain cell, the pathogen steals the nutrients from the infected cell, leading to a decrease in energy availability. Instead of actually having to notice the pathogen, the cell can use its own energy metabolism as a marker for infection and act accordingly with an immune response. The cell responds by shifting the amyloid metabolism towards the production of longer, more potent antimicrobial peptides that can aggregate in plaques that halt and arrest the pathogen, toxic oligomers that can kill the pathogens, and pierce the blood-brain barrier, which is a feature of Alzheimer's disease called cerebral amyloid angiopathy, in order to allow the entrance of specialized immune cells in the brain. Increased phosphorylation of tau protein leads to the intracellular formation of tau tangles that can capture and kill the pathogen. This actually also prevents disease spread by disrupting the cytoskeleton, as we know that pathogens can use the cytoskeleton to move inside the cell. So the infected cell is sacrificed in order to save the healthy cells. Now let's apply the same mechanism to the brain of an aging person with multiple risk factors for Alzheimer's disease. In this scenario, the energy metabolism of all cells start to decrease, potentially to the largest extent in the areas of the brain that normally use the most energy, such as the cortex and the hippocampus. Now, since the brain cells are evolved to interpret this energy decrease as potential infection, the same mechanism leads to the onset of a huge, out-of-control protein aggregation reaction and neuroinflammation, which is followed by progressive cell death and what we call Alzheimer's disease. So to recap, aging and Alzheimer's disease risk factors decrease the cerebral energy metabolism, which the brain cells may interpret wrongfully as an infection. The cells respond by increasing the production of more potent antimicrobial variants of amyloid and tau protein, leading to excessive aggregation, neuroinflammation, cell death, and ultimately Alzheimer's disease. Note that there are, of course, genetic factors such as carriage of the ApoE4 gene variant that may directly influence protein aggregation or other processes that increase the possibility of developing Alzheimer's disease. So in this scenario, what can we do to counter the process? The ideal intervention would be one that maintains adequate energy metabolism in all brain cells during lifetime. That way, the process wouldn't start in the first place. But since this is not realistic, the next best option is to optimize the cerebral energy metabolism and to reduce the immune response. To optimize the energy metabolism, we need to combine interventions that increase both the supply and the demand of energy in the brain. It doesn't work to give food to someone that is not hungry, and a hungry person cannot be helped if there's no food available. Consistent cognitive and physical activity can be combined with dietary interventions and medications that stimulate brain energy metabolism to create a synergistic effect. Then, if there's no infection, the immune response needs to be halted. Cells do not function properly under inflammation. We need interventions that counter amyloid and tau aggregation in combination with anti-inflammatory drugs that reduce neuroinflammation. These therapies already exist, and many have already shown promising results in clinical trials individually. But if the disease process has already begun, the individual treatments may not be sufficient. And just like in HIV, it is very likely that a cocktail of treatments is necessary in order to halt the disease progression. And in order to select the right interventions, we need to reach consensus on the driving force behind Alzheimer's disease.